All right, and welcome again, everybody, to the interactive review session in preparation for our 2025 local anatomy competition. That is set to be held in just under 2 weeks on January 25th, 2025. And qualifying students from that level of competition will be invited to their respective regional competition. More information about that regional competition will be sent out onto our website and emailed out to all of you guys shortly. Practice questions can now be found on our website in addition to this interactive review. This is going to be underneath the resources tab, and there's now a new page called practice questions. These were made by our Northern Regional winner from last year, Michael Harkins, and medical students at Drexel who helped compile the images for the image-based practice questions. So those questions are different from the ones tonight, and we highly encourage you guys to take those in addition so that you can get a sense for where you are in your preparation. Just as another reminder that registration officially closes tomorrow. That's the 13th of January at midnight Eastern time. So if you guys have any friends that were last minute wanting to hop on board, the time is ticking on that, and tell them to get signed up, and we'll be happy to have them. The local is launched across all time zones at the same real time for the local competition. So you will be testing at the exact same time. So me in Alabama are going to have people testing at the exact same real time as those of you that are testing in California, for example. So the start time for Pacific Standard Time will be 9 a.m., 10 a.m. for Mountain Standard Time, 11 a.m. for Central Standard Time, and 12 noon for Eastern Standard Time. Because we were trying to maintain the integrity of the exam, all start times are set and you cannot change times to anything else. So if you have any conflicts of interest or anything that you can't attend that specific time that is set, unfortunately, we won't be able to have you participate this year. But if you do miss this year, we do encourage you, if you're eligible in future years, to still sign up and have you participate in an upcoming competition. You should receive a link from your local host chapter on or by the 17th of January, that is this Friday, regarding any of your participation for this year's examination. So please keep an eye out for that email from your local host chapter. If you don't know your local host, that's okay. We've paired them for you, so you don't need to worry about that. If you didn't get an email by that date, then please feel free to email us and we'll make sure that you get access to that. Just a final reminder is that the certificate of completion for the local competition, which can be verified with a unique code for you to submit on all of your application materials, that's something that we provide to all those that compete in the local level of competition, regardless of score. So again, we highly encourage you, regardless of what your score is, to come show up and participate and put this on your CV when you guys are getting ready to apply to your undergraduate institutions. So those are all the updates that I had. Anything else from our Anato-Bee?
Yes, Dr. Proskura, there's a question in the chat that I think would be really important that we just on the recording answer. So do you see it there?
I do, yeah. Where's the link to register to take the exam? So you should already be registered. The fact that you are here, you've already been acknowledged that you're registered to take that examination. The emails that we provide these tutoring links to are taken from that registration process. So you should already have that registration done if you're on this Zoom link. If you haven't already and you're not attending this live Zoom, then you will need to go on our website if you weren't getting those links and register with an email that can accept emails from external communicators. So some of your high school emails block external communication. So if you haven't gotten these emails and you're wondering why there's all these other people that have been able to attend and you've just been missing those, but you registered, please just re-register with an email that can receive those communications, and that should resolve that issue. So you are registered to take it, and then the information that you will need to take that exam again will be emailed to you from the local chapter coordinator that we've provided your information to. So you don't need to do anything else besides keep studying and wait for that communication. And then if you don't, feel free to reach out to us.
And just to clarify, you're not going to receive an actual exam link until like the day of the exam or the day before. But even if we decide to send the link the day before, you will not be able to access it until the time of the exam. We don't open the exam until the time you are given by your local chapters. So if you're thinking you didn't receive the exam link and you're missing something, you're not. It's just we don't give that information out until it's actually time to take the exam.
Yeah, thank you for that clarification. So you'll receive a Zoom link, and then on that Zoom link, you'll receive also, we'll have that chapter coordinator put that in there too. So thank you for that clarification. Any other questions or anything before we get started? And all righty. I see. Time-wise, how long is the exam? So you'll be given 2 hours in total. You can hop on that Zoom link. It should open 30 minutes before the exam start time. So if you allot about 2.5 hours total for that exam, and then you don't need to stay until the end of it. If it doesn't take you that long, you can go ahead and submit your exam and log off of that Zoom link. So you won't need to stay for that duration if you finish your exam.
And again, just maybe a reminder, it's 150 multiple choice questions. 100 of those are just written, and then we have 50 of the image-based multiple choice questions. And again, the practice exam should give you a really good idea of what the format is going to look like and the difficulty level of the questions. The exam for the virtual local competition is going to be very similar, just longer in length than what we've posted on the website as a practice exam.
All right, with that, we'll go ahead and kick this off and start getting into our review session materials here. So first, I believe normally is our moderator, and so normally he's the one doing the introductions for all of you, but I'll go ahead and just at least acknowledge that Bill Frank has been our moderator for all of our tutoring sessions since we started these tutoring sessions even last year, and he's been here helping answering questions in the chat, so many of you guys are familiar with him already. He is a faculty member at Drexel University along with Dana Peterson. And we've just really been thankful for all of his time here. And he's got some really good questions prepared for you. So thank you, Dr. Frank. If you want to go ahead and take over.
Okay, well, thank you very much. It is a little different being on the other side, being the presenter instead of the moderator. First of all, I wanted to thank Dr. Pascura and Dr. Peterson for their assistance in creating this review slide deck. So I wanted to give that acknowledgement first and try to get my screen shared here. Okay, and everyone can see that just fine. Okay. All right. I did want to, as before we start question one, I think I accomplished this, but at the bottom of each question, you can see the learning objective where this question was created. So with each question that I go over, you'll be able to tell later on where that actually came from so that you can go back. If you're not sure of what the answer is, you can go back and find where, which session that was a part of. So, okay. So, Dr. Pisker, can we have the first poll? So go ahead and answer question one. And
could you read this question aloud for us, Dr. Frank?
sure. Which of the following organ systems and functions is paired correctly? So Dr. Piskura, whenever you feel it's good to close the poll, please do so.
You guys are doing a really good job with this. I'll go ahead and close this poll in 5, 4, 3, 2, 1.
Okay, so we're looking at the organ systems and functions, and of the answer choices, the one that is paired correctly is the skeletal system, and the skeletal system supports the body and enables movement. So the other four are not paired correctly, and I can go through fairly quickly. Remind you that the integumentary system encloses the internal body structures. It also is the site of many sensory receptors. We know that the skeletal system supports the body. The endocrine system secretes hormones and regulates body processes. Cardiovascular system delivers oxygen and nutrients to tissues and equalizes temperature of the body. Muscular system is involved in movement and helping maintain body temperature. Nervous system detects and processes sensory information, and activates bodily responses. Lymphatic system returns fluid to the blood, that interstitial fluid that escapes the cardiovascular system will eventually drain back into the cardiovascular system through the lymphatic system. Respiratory system removes carbon dioxide from the body and delivers oxygen to the blood. Digestive system processes food used for the body and removes waste. Urinary system controls water balance in the body and removes waste as well. The male reproductive system produces sex hormones and gametes, and the female reproductive system produces sex hormones and gametes and supports the embryo and fetus until birth, and also produces milk for the infant. So it would be important to have a general understanding of each organ system and function. Okay. Let's go to question number 2. Which of the following is pointing towards the dorsal aspect of the body? Can we get a poll?
Yes, I'm sorry. I realize that this one doesn't have the right number of. So for this one, I'm going to say this one's launched, but if you would like to put any options that aren't in there, I think it just goes to F,
Okay.
G, or H. You guys can type them in the chat,
Okay.
and I'll fix that for the ones moving forward. All right, we'll go ahead and close this poll in 5, 4, 3, 2, 1.
All right. And it looks like majority of people chose the right answer, and that is actually D. So if we go to the next slide, we can see the anterior and posterior parts of the body are also referred to as ventral and dorsal. And if you remember, the thoracic spine is sometimes called the dorsal spine. Lateral and medial. Medial is going to be towards the midline of the body, where lateral is going to be towards the side of the body. Proximal and distal. Proximal is going to be more towards the origin or center of the body, and distal is going to be further away from the origin or center of the body. Lastly, superior and inferior also go by the terminology of cranial, or towards the head, and caudal, or towards the tail or the inferior portion of the body. Okay, so very good. Okay, so for body planes, which plane would divide the body into an anterior and posterior aspect?
All righty, we'll go ahead and close this poll in 5, 4, 3, 2, 1.
Okay, so about a little over half of you got the right answer. And the correct answer is the coronal, also known as the frontal plane. So in the chat, can you tell me which of these is the coronal or frontal plane? Is it 1, 2, or 3? Okay, I think most people said 2. So the frontal or coronal plane is the plane numbered 2. And we know that the midline or the sagittal plane divides the body into right and left halves. So if it's in the midline or the mid-sagittal plane, it is going to divide them equally into right and left. But they could also be left or right of midline. So that would be a paramedian or a parasagittal section. They're still going to divide into right and left halves. They just won't be even. The frontal or coronal plane divides the body into anterior and posterior. And lastly, the transverse or horizontal plane is going to divide the body into an upper and lower section. Okay, so very good on the chat. Okay, so question number 4. Which of the following indicated the movement of the arm away from the midline of the body?
And this poll will close in 5, 4, 3, 2, 1.
Okay, well, majority of people got the right answer. That is D, abduction. So abduction is the movement of the arm away from the midline of the body, bringing the arm back towards the midline of the body as adduction with 2 Ds. So abduct is going away, adduction is coming towards the body. Circumduction is a circular motion. I don't have here, but pronation and supination are dealing with the upper extremity. Remember, the palms facing forward is anatomical position. And so if you move the hands internally, or like putting palms down, that's pronation. And if you bring the hands back to where the palms are facing forward, that is supination. Okay, so pronation, supination. All right, good job. Did I tell you that so far you're doing really, really well? And I'm super happy with where you're at. So keep up the great work here. You're all doing very, very well. Not surprised, but but very happy. So question number 5. The dorsal body cavity contains which major organs? It seems to be a little.
All right, this poll will close in 5, 4, 3, 2, 1.
Okay, well, majority of the people chose A, brain and spinal cord, and that is the correct answer. So the dorsal body cavity is basically the skull and the spinal cord, or the spinal column, which contains the brain and the spinal cord. So all of the nervous tissue that's sitting inside of the bony cavities is part of the dorsal body cavity. In the anterior body cavities, we have 3 different subsections. The thoracic cavity, which is separated from the abdominal cavity by the diaphragm, is part 1. in part 2. And then the third part, which is not divided by a diaphragm or a partition, it's just an area of transition between the abdomen and the pelvic. So sometimes that area is called the abdominal pelvic cavity. Okay. So very good. And obviously, thoracic cavity is going to contain the heart and lungs. Abdominal cavity is going to contain the majority of the gastrointestinal organs. And then the pelvic cavity is going to contain the bladder and sexual organs. Okay. So good job. Okay, question 6. Moving right along. Which of the following vertebrae is being indicated by the arrow? If you can't see the arrow, it is right there. So what is this area?
This poll will close in 5, 4, 3, 2, 1.
Oh, there are a few last minute changes. Okay. A good point to uh, indicate is that sometimes your first impression is the correct impression, right? And so there looked like at least one person that changed their answer right at the bell, but they were actually correct. And the area that this is pointing to is the lumbar spine. Remember that the uh, affiliated with the skull is going to be the cervical spine, which has 7 vertebrae. This next area is going to be the thoracic spine with 12 vertebrae. And then the third section is going to be the lumbar spine with 5 vertebrae. The ending of the vertebral column is going to be the sacrum, which is 5 fused vertebral segments. And lastly, the little tail here, the coccyx, is made up of approximately 4 fused segments. Okay, so cervical, thoracic, lumbar, sacral, and coccygeal segments of the vertebral column. So very good. Okay, good job. Question 7. Which of the following is classified as a synovial joint? Okay, so which of these is an actual synovial joint? Okay,
Right, this poll will close in 5, 4, 3, 2, 1.
okay. Well, majority of you were right on. And the hip joint is actually a synovial joint. It's a ball and socket joint, actually. And it is lined with cartilage, and it has a capsule. And inside the capsule is synovial fluid, which helps it move. The suture of the skull are fibrous joints, and the intervertebral disc, the rib cage, the cartilage, and the pubic symphysis are all cartilaginous joints. Okay, so is the hip a part of the axial or appendicular skeleton? Can you put that in the the chat box. Is the hip actually part of the axial or appendicular skeleton? Okay. Right, so great. Most are saying it is part of the appendicular skeleton. It is kind of that transition between the axial skeleton, which includes the skull and the ribs and the sternum and the vertebral column, the sacrum and coccyx, and the transition from the pelvis and to the lower extremity. So great job. Great
And
job.
I do want to say, Dr. Brink, if I could just intervene for just 1 second. I will say C is partially correct.
There are some
There's some synovial joints, and
synovial.
I hesitated to bring this up, but I wanted to mention it only because if you think about synovial helps create movement. So to help movement while we breathe, we need to have synovial joints in that anterior chest wall. And that's the only reason why I brought it up. But that's
No
also
problem.
into very nitty-gritty details of our tutoring session as well. But you all are doing a really great job. And for the overall consensus, this is correct. And I think, did we have a question pop up in the chat? Oh, everybody's answering. Yes. Does the hip joint have a bursa? What's the question?
The bursa are associated with areas of bony prominence and are usually located around joints. There are multiple bursa that are located around the hip, at the greater trochanter, at the ischial tuberosity, which is what I call the butt bone, where you sit, that bone that you feel. So the bursa have a role of providing decreased friction between the soft tissue that's either attaching to or around those bony prominences. So it is usually affiliated with joints, but it is not part of the joint capsule or part of the cartilage of the joint. All right, let's move on to question number 8 here. And I think we might need to speed up these questions a little bit, Dr. Pascura. What is the role of synergistic muscles in muscle function? So what do synergistic muscles do, or synergist muscles do?
All right, and we'll go ahead and close this one in 5, 4, 3, 2, 1.
Okay, majority of you said C. Synergists contract to stabilize a joint to allow an agonist or a muscle that's working in conjunction with the other muscles. It allows it to work more effectively. So we can see here in this example, in raising a glass up towards your mouth, we would want the brachioradialis and the brachialis muscles to stabilize the elbow. So as the bicep brachii flexes the shoulder, that we're able to do so, but they're working in conjunction with each other to create one motion, which would hopefully be to drink that cup of orange juice that is in the cup. Okay, good job. Okay, let's move on to question 9. Okay, so which of these muscles abducts the shoulder joint when it contracts?
All right, and this poll is going to close in 5, 4, 3, 2, 1.
Okay, excellent. Majority of people said the deltoid muscle. The deltoid muscle is in the lateral aspect of the shoulder, and when it contracts, it's going to bring the arm away from the midline of the body. So it is an abductor of the shoulder. There is another muscle here that abducts, and that is going to be the supraspinatus muscle, which is one of the four muscles of the rotator cuff. And so it actually helps in the early part of abduction of the shoulder, and then the deltoid takes over. Okay, so good job. Right. Let's go to question 10. In what anatomical structure of the nervous system would you find collections of sensory neuronal cell bodies? So sensory nerve cell bodies, where would you find them? In what of the following structures?
Alrighty, we'll close this poll in 5, 4, 3, 2, 1.
Okay, this one was a little tougher. Okay, so the correct answer is ganglion. And we talked about in this little view here, we can see this is a cross section of the spinal cord, and associated with the spinal cord are the dorsal root ganglia. And that is where the cell bodies of the sensory neurons for the peripheral nervous system are going to be located. This is for the somatic system. Okay, so the cell bodies of those pseudo-unipolar neurons are going to be located in the dorsal root ganglia. Okay, so an afferent neurons carry sensory information from the periphery to the central nervous system, and the cell bodies are located outside of the spinal cord in the dorsal root ganglion. So if I can get that to, okay, so we can see that coming into the spinal nerve, dorsal root ganglion, and then it's going to project a central process to the dorsal root of the, or the dorsal horn of the spinal cord. The efferent neurons are the actual motor neurons. They carry motor information from the central nervous system to the peripheral nervous system. So cell bodies can be located in two parts. In the autonomic system, they're going to be located in the lateral horn of the anterior horn of the uh, gray matter, and those are going to project to ganglia outside of the central nervous system, and then eventually to the organs, and these are for involuntary motor responses. We also have the somatic system, which is used for voluntary motion. The cell bodies are in the anterior horn, and they're going to go directly to the muscle cells. And so this is a one neuron system, sometimes called the final common pathway, because once the neuron is stimulated, it has one job to do, and that is to cause contraction of the muscle. Okay, so understand the difference between afferent and sensory nerves versus efferent or motor nerves, and understand efferent motor nerves have either an autonomic system or a somatic system. Okay, right. Let's go to question 11. Which of the following best describes the functionality of the central nervous system?
Right, and we will close this poll in 5, 4, 3, 2, 1.
Okay, excellent. So the central nervous system function is to process and integrate information. And so we know that that is the process of the central nervous system. The peripheral nervous system is going to bring information back into the central nervous system and also transmit information away from the central nervous system. The somatic nervous system is involved in voluntary actions and is both somatic motor function as well as somatic sensory function. So basically, you move your hand over a hot stove, you can sense that, and it's going to send that to the central nervous system, which is going to tell you that that's painful, and the central nervous system will process and tell you to move your hand off of the stove. Okay, so this is a voluntary system. The autonomic nervous system is used in involuntary functions. Okay, and so they have autonomic motor as well as sensory fibers as well. There are 3 different types of autonomic nervous system functions. One is the sympathetic nervous system, which is also known as the fight or flight uh, response, and that's going to do things like increase heart rate, decrease digestion, and cause the production and release of adrenaline. The parasympathetic system, known as the rest and digest or rest and repose, is going to usually have the opposite effects of the sympathetic system. So it will do things like slow the heart rate, but it will increase digestion. And lastly, the enteric nervous system is contained within the wall of the gastrointestinal system, sometimes called the body's second brain, and it is going to control the smooth muscle movement of the intestines, but it will also help with blood flow and secretions of the gastrointestinal system. So the autonomic nervous system has 3 different sections. It would be very important for you to understand the general processes of each of those systems. Okay, question 12. After oxygenated blood leaves the lung, which chamber of the heart does it enter? Okay. Where is the blood returning to the heart after it's received all the oxygen from the lungs?
All right, we'll go ahead and close this poll in 5, 4, 3, 2, 1.
Right. Excellent job again. Majority of you got it right. The left atrium. Remember that the deoxygenated blood is coming from the inferior and superior vena cavas into the right side of the heart, so the right atrium, and then the right ventricle. The right ventricle will push that blood to the lungs to become oxygenated. So when they are full of oxygen, they're going to return to the heart via the pulmonary veins to the left atrium. Then it will be pushed into the left ventricle and then eventually up the aorta and to the rest of the body. So it's important to understand the flow of blood, both deoxygenated and oxygenated blood through the normal adult heart. Okay, job. So this is more of a identify here. Which arrow is correctly identifying the right ventricle of the heart? Which is the right ventricle?
Dr. Frank, you have a question in the chat about whether or not they're looking at the anterior or the posterior view. So maybe you could just give them a few hints on that.
Okay, yes. So we are actually seeing the anterior view of the heart. We can see the right lung and the left lung. We can see the brachiocephalic veins coming in and creating the superior vena cava. Okay.
All right, we'll close this poll in 5, 4, 3, 2, 1.
Okay. Okay. So the correct answer is B. B is the right ventricle. We can see that here in the cartoon. I did want to point out that the majority of the heart that is up against the diaphragm, the central tendon of the diaphragm, is the right ventricle. And so A is the right atrium. D is the left ventricle. And C is actually the pulmonary trunk, which is the outflow of the right ventricle. And the one that wasn't identified, that is the aorta. We can see the right coronary artery and a branch of the left coronary artery, which is the left anterior descending artery. And this is the apex of the heart. So if you think of the heart kind of reclining on this on your right side, the right atrium is kind of your arm and upper body off the floor, and the hips and legs are the right ventricle. So if you can think of it like that, the right ventricle is most associated with the diaphragm. Okay, let's go to the next question, 14. What is the function of the pulmonary arteries in the respiratory system?
And this poll will close in 5, 4, 3, 2, 1.
Okay, so the correct answer here is transportation of blood. Okay, so the pulmonary arteries are going to transport the deoxygenated blood up through the pulmonary trunk and then to the left and right pulmonary arteries to the lungs. Okay, so pulmonary arteries are going to transport deoxygenated blood to the lungs. Okay, so question 15. Identify the structure indicated by the arrow. So you can see here. So this is an anterior view, and we're actually looking at the mediastinum of the right lung and the mediastinum of the the hilum. I'm sorry, not mediastinum. Hilum of the left lung, the hilum of the right lung. So what is this structure here indicated by the yellow arrow?
All right, and this poll will close in 5, 4, 3, 2, 1.
All right. Excellent work again. So this is the trachea. Remember, the trachea begins at the end of the larynx, and it is going to come down through the superior mediastinum into the posterior mediastinum and approximately the level of the sternal angle of the chest wall. It's going to break into a right and left primary bronchus. And so internally, if you look down, you could see an area known as the carina, which is the bifurcation of the right and left primary bronchi. So very good. Okay, let's go to question 16. Which of the following structures contributes to both the foregut and the midgut?
Yeah, we'll close this one in
Okay.
5, 4, 3, 2, 1.
Okay. Okay, so the correct answer is the small intestine. We know that the foregut is going to include the distal part of the esophagus, the stomach, and the first two segments of the duodenum, which is part of the small intestine, along with accessory organs such as the liver, the gallbladder, the spleen, and the pancreas. Okay, so I've included this slide of the differences between the foregut, midgut, and hindgut, including the organs, the arterial supply, and the function. And the arterial supply is helpful too, because that is used in embryology as well to help separate the three divisions. Okay, so that's for your reference. I also added this as well. These are slides of the celiac trunk, which supplies the foregut, and then the superior mesenteric artery, which supplies the midgut, and then the inferior mesenteric artery, which supplies the hindgut. So again, these are for your references to help you understand the separation of each of those. Okay, so we'll move on to 17 here.
So Dr. Frank, it's
Yeah,
Dr. Peterson. Maybe for the
yeah.
sake of time, I know we don't typically post the PowerPoints, but I know that you're really good at making these recordings and editing them. So maybe could we stop here? And would it be possible that you could just finish up sort of a narrated recording that you could send to Dr. Piskura that she could post up on our website, just finishing out the slides? Because I really want
Sure.
all of our participants to be able to hear your narration and the explanation. But I think really we should probably think about moving on to the histology slides.
Yes, I agree.
I just also wanted to address, or Dr. Frank, you could address, there is one question in the chat that I would like one of the faculty to answer. I see that one of the participants had answered for another participant asking the question, but I think there needs to be a little bit more clarification in the answer. So it was, what's the difference between an atrium and a ventricle?
Okay, so if you think of a house, the atrium is the entrance part of the house, usually right inside the front door. And usually there's a chamber area. And so that's what the atria are. Those are the entry areas into the heart. So the right atrium is getting the deoxygenated blood from the body that's entering the right side of the heart. The left atrium is receiving all of the oxygenated blood from the lungs and receiving that into the upper part of the left side of the heart. So the ventricles are going to be the lower chambers. So those are the muscular chambers. They're thicker. They're going to, when they pump, they're going to be pushing out to either the lungs on the right side, or in the left side, they're going to be pumping really to the rest of the body. Okay, so that is the difference between atrium and ventricles.
Thank you, Dr. Frank, for all that.
You're welcome.
Yeah, really appreciate that great presentation, helping us walk through those questions, and looking forward to more that you'll help us with in addition to that. I can say last year we didn't make it through all the content in the 2 hours that we had planned for either. So we just have so much great stuff that we want to share with you guys. And but we'll go ahead and move forward then. So it's my pleasure to introduce Dr. Janelle Mapes to talk about all of our awesome stuff in histology. She is an absolute rock star when it comes to histology and anatomy and embryology as well. But she just is a content expert in this. She's a faculty member at the Pacific Northwest University of Health Sciences. She came on with the Anato-Bee as a regional host for the Western competition last year. And not only did she help make sure that that competition was successful, but she helped with producing content that made sure that all of the regional competitions were a success last year. So we're incredibly grateful that she's joined on with us and that she's been helping us with additional stuff and joining the Anato-Bee team this year. So without further ado, Dr. Mapes.
Thank you, Dr. Piskura. What a lovely introduction. Hopefully I can live up to your description there. So folks definitely brought their A game tonight when I'm seeing the results of the anatomy review portion. So really excited to jump into the histology. So in terms of the learning objectives, those are on the website. I wish that I saw that Dr. Frank put them at the bottom of the slide because that would have been great for me to do, but I guess that's always for next time. Thanks always as Dr. Frank helping us learn new things. All right, so we're going to start with this first question. Identify the epithelium in the image.
All right, we will go ahead and close this poll in 5, 4, 3, 2, 1.
Yeah, still looking good. So what we're looking at here is a stratified squamous epithelium. So we've got multiple layers, which makes it stratified. So if it was just one layer, we'd call it simple. More than 2 layers, we'll call it stratified. And the key thing here that we're looking at, right, is that we're looking at those surface cells, that most apical layer of cells. So facing the lumen here would be our apical side. Basal side is facing the underlying connective tissue. So we want to look at these cells up adjacent to the lumen or the white space, or external. If this was skin, it would be just the air, right, surrounding the body that you'd be looking at for that apical layer of cells. All right, so next question, what is the function of the epithelium in the image?
All right, you guys are doing good with these histology questions. We'll go ahead and close this one in 5, 4, 3, 2, 1.
Great job, everybody. So we're again always thinking about structure and function in histology. I've been told by my medical students that they literally have nightmares about me talking about structure and function. So thinking about all of these layers of cells, right? If you have something that scrapes this top layer, you're still really well protected by these underlying layers of cells. So that's the perk of having this stratified epithelium. So let's take a look at all the different types of epithelium. So like I mentioned, simple would be one single layer of cells. Stratified is going to be multiple layers of cells. We also see shape differences, right? So squamous is going to be the flat kind of fried egg shape. Cuboidal are cube shaped, right? So they're pretty much as tall as they are long. That simple columnar epithelium, so those column shaped cells. Pseudostratified columnar epithelium is one of those weird ones that it looks like it's multiple layers, but the key thing is that all of them are going to come down and touch this basement membrane. So once you start adding additional cell layers, the cells at the basal side can start to look kind of weird. They can start to look like different shapes. So in that last picture that I showed you of the stratified squamous epithelium, if I go back, these almost look cuboidal, right? The nuclei look pretty round. They look like they might be square, right? So that's why we're always going to be looking at that top layer, that apical layer, to help us figure out what type of epithelium that we're looking at. So here again, we're going to pair that structure function. So in simple squamous, places that we're going to see that are the alveoli of your lungs, because they're really, really thin, really easy for gases and nutrients, all kinds of things to diffuse back and forth between these. It's also going to be like lining the outer surface of a lot of your organs as well as a serosa that helps with kind of gliding. So most of your digestive tract is going to be lined by this simple squamous epithelium to help for reduce friction in that movement. Places that you have a simple cuboidal epithelium would be like your kidney, really good for secretion and absorption. So as your kidneys make urine, we're going to be moving ions in and out and all kinds of things. So this shape is really helpful for that. Simple columnar epithelium, we're going to find those places like the lining of your digestive tract and the respiratory epithelium. These cilia are the more apical structures. You can have cilia in the respiratory tract that beat to move mucus, any particulates that get trapped in there. You can also have microvilli here, lining these that provide more surface area for absorption in the digestive system. So just stratified columnar epithelium, we're also going to see this in the respiratory systems, mostly with cilia. So again, helpful for trapping mucus and particulates. Stratified squamous epithelium, we're going to have in places that are going to be kind of in contact with abrasive substances. So things like your mouth and the esophagus right before the food gets broken down. I always think of like chips as being like the most destructive thing that you can put in your mouth, and I don't know why. So that stratified squamous epithelium, right, is going to protect from any pointy chips that might be trying to tear up your mouth and your esophagus. Stratified cuboidal is also kind of protective. This is pretty rare. We'll see this mostly in ducts. Same thing here with the stratified columnar epithelium. Once those ducts get bigger, that's when we start seeing this. And then transitional epithelium, also called urothelium, is also going to be found in the urinary tract. And so this tissue is really cool in that it's able to expand and contract. So as your bladder fills, right, the musculature and all the other connective tissue there helps to allow that to stretch, but the epithelium is going to need to move as well. There's also this really cool protein on the apical surface of these cells called uroplakin that helps protect these cells from any damage from urine. So the outer layer of any organ, so it depends. So we have organs that are intraperitoneal, and then we have organs that are retroperitoneal. So we think about intraperitoneal organs, those are going to be the ones that are kind of like, I think about they're like loosey-goosey inside like the abdominal cavity. So if you think about your small intestine, right, it's going to be kind of moving around. And then we have some organs that are really kind of adhered in place and connected well to one another, and those are going to have an adventitia. And there's a connective tissue that just kind of blends with one another. So if you were to take a section through the mediastinum and you looked at the difference between the esophagus and the trachea sitting right next to each other, it's going to be hard to figure out where that outer layer of adventitia of the trachea ends and where the outer layer of adventitia of the esophagus starts, just because they kind of really blend together. So in some of these organs where they're really kind of more stuck, that's an adventitia, but ones that have a serosa, they're intraperitoneal, yes, those are going to be lined by a simple squamous epithelium. Great. All right, so let's move on to our next tissue type. So what is the primary function of fibroblasts in connective tissue?
All right, we will close this poll in 5, 4, 3, 2, 1.
Fantastic on this one. So this is going to be producing fibers and ground substance. So we can collectively refer to these as extracellular matrix. So we can be making collagen fibers or elastic fibers. Ground substance is kind of like this viscous, gooey gel that helps us hold this connective tissue together. So the fibroblasts are the resident cells here in connective tissue. You can find them in multiple different types of connective tissue. I also want to just let you all know how impressed I am that you got this question right. I know some medical students that wouldn't know the answer to this question, so you're doing fantastic. So if we look in these other options here, so store excess lipid molecules, that'd be function of an adipocyte. So an adipose tissue, engage in an immune response. So those would be our white blood cells of the immune system, and then carry oxygen and carbon dioxide. Those would be the red blood cells in the blood, or erythrocytes. All right. So moving on to the next question, identify the tissue in the image. And we're looking at just this middle strip right here. It's a good opportunity to remind you that organs are going to have multiple different types of tissue, right? So when we take a section, you'll see some other tissue types in there as well. So we're just looking at this strip here in the middle.
All right, we will close this poll in 5, 4, 3, 2, 1.
Thanks. So here, this middle strip that we're looking at is hyaline cartilage. So we've got a perichondrium, so that's a connective tissue that's going to be on either side of that cartilage. And then we can see some chondroblasts, are the cells that are going to be building that cartilage matrix. And then the chondrocytes here trapped in their little lacunae in this hyaline cartilage. You can see on the slide as well, there's some adipose tissue down here, and then some skeletal muscle up here as well. All right, so just a quick review of hyaline cartilage. So this is by far the most common type of cartilage in the body. It's really good at what it does. A lot of times you'll find it at the end of bones and joints. So we're going to have this articular cartilage, which is made of hyaline cartilage to help prevent your bones from grinding up against each other in those joints. You can find it in your nose, so the tip of your nose, your rib cage. So on that anterior portion where those ribs are connected to the sternum, that's all hyaline cartilage. Those tracheal rings that Dr. Frank was just pointing at in that gross anatomical section, those are also made of hyaline cartilage. So hyaline cartilage is going to be in lots of different places. When we're looking at the differences between these three different types of cartilage, I always think of hyaline cartilage as kind of like the standard. This is my basic type of cartilage. And then for the other two types, I feel like we're just adding other types of fibers or other type of extracellular matrix. So here we've got this cartilage matrix. If we add collagen fibers to the cartilage matrix, it's fibrocartilage, right? So we still have the chondrocytes and the lacunae, but then we have all of these collagen fibers here as well. In elastic cartilage, we're adding elastic fibers, okay? So here, this kind of dark pink is where you can see those elastic fibers there in that matrix. So all three of these are going to have chondrocytes, they're going to have chondroblasts, but it's the differences there in that extracellular matrix that are going to give it slightly different function. So like fibrocartilage is going to be a little bit more stiff and strong because of the collagen fibers there in that matrix. And then elastic cartilage is going to be a little bit more elasticy, right? So like your ears have elastic cartilage, right? So if you bend your ear down, it's going to flop right back. That elastic cartilage is really what's helping it do that. So again, always structure function. All right, so moving into skeletal muscle, which part of the sarcomere only contains thin filaments? And I gave you this picture here just to give you a little bit of help. Like this one's a little bit more of a challenge. It's good.
Okay, we will go ahead and close this poll in 5, 4, 3, 2, 1.
All right, so this portion of the sarcomere that only has thin filaments is going to be the I band. So let's move on to the next slide. I think give you a little bit more images. So when we talk about thin filaments, we're talking about the actin. And so that's what's green here as well as down here. And then we're talking about the thick filaments, we're talking about myosin. That's purple in both of these pictures as well. So when we lay out the sarcomere, what we're going to do is go from Z disc to Z disc, and that makes our full sarcomere. Here, that Z disc are going to have proteins that are going to be important for attaching these actin filaments to one another, provides a really nice anchor point for these filaments in adjacent sarcomeres. Okay. So within, if we expand out, right, this Z disc, what we have is the I band. Okay. So then that I band is only going to contain actin filaments. Okay. If we look at the other structures here, we have the A band. So the A band is this large segment that's going to cover all of the thick filaments. Okay. So you can see here at some point we do have some overlap, right, between thick and thin filaments, and that's going to change whether or not this muscle fiber is contracted or relaxed. Okay. So here we have this relaxed muscle fiber. The sarcomere is stretched out, so there's not very much overlap between these thin filaments and thick filaments, but down here in this image, you can see there's a lot of overlap, so it's more of a contracted. The H zone is the portion that only has thick filaments, so no thin filaments here. So again, it's going to be a little bit different size in this relaxed versus contracted. And then we have this M line down the middle that I'm just realizing now isn't labeled on any of these pictures, but this would be the M line. And that's going to anchor all of these thick filaments, right? So these myosin thick filaments have these little heads. And so when ATP causes movement of these heads, they're going to grab on to these thin filaments and pull them closer. And that's what's going to move these actin filaments actually closer here to this H zone as that fiber contracts. Cool. All right. So in looking at the different types of muscle, so what we were just looking at is skeletal muscle. So here we've got multiple cells that fuse together into one muscle fiber, or we can call it a syncytium. And so we've got multinucleate cells because those individual cells have fused together. So these are going to be really long. They're not branched, and where we're going to see the nuclei is at the periphery of the cell because they're really packed with all of this actin and myosin that's arranged in those sarcomeres in a very particular way. And that's going to produce these striations, and that's going to allow, right, these skeletal muscle cells to contract. They're under voluntary control, so we've got motor neurons, right? They're going to tell our skeletal muscle to contract, and where we're going to find these is the big muscles that we are identifying in anatomy. They are made up of skeletal muscle. Then we have smooth muscle. And so these cells are a little bit different shaped. They're more spindle shaped. They only have one nucleus, and it's centrally located, as opposed to this peripheral location of the nuclei here in the skeletal muscle. They still have actin and myosin, but they're not like pre-assembled into these very organized sarcomeres. They're going to more or less assemble when there's a stimulus for them to do so. There's lots of gap junctions between these cells so that we can get really good communication of ions. So we get this kind of contractile wave. They're under involuntary control, so you're going to find them in places like your organs, right? So that enteric nervous system in your digestive tract is going to help control contraction and relaxation of the smooth muscle. They're also going to be kind of like tonically contracted to hold. So like your arteries are going to have a lot of smooth muscle in their walls, and that's really going to help them hold their shape. And then the last type of muscle here is cardiac muscle. And so again, it's kind of a functional syncytium. You've got maybe like a couple cells that are going to be connected, but they're connected via these intercalated discs. So there are these lines here, and then pointed out with these arrows here. And so these discs really help to anchor the cells to one another, but they're also going to allow communication. Again, those ions flowing back and forth, telling them to contract. So we've got these branching cells. Nuclei are essentially located. They're also striated. So we also have this kind of pre-organized actin and myosin. It's under involuntary control, so the place that we're going to find cardiac muscle is in the heart, and then the first parts of those great vessels. All right. So moving into neuro, what is the most common type of neuron? I've got a question in the chat. Are there disorders caused by actin not pulling on myosin? So it's actually the opposite. It's the myosin that pulls on the actin. One thing that's I think is kind of cool, maybe a little bit weird, when you pass away, you're not making any more ATP, and you need that ATP for the myosin to actually release from the actin. And so that's why people have like tonic contraction of their skeletal muscle shortly after passing, because the myosin, it just isn't going to let go of that actin. Eventually, as the cells die, the lysosomes break open, and you have digestive enzymes that are kind of wreaking havoc on the body, and so those skeletal muscle fibers eventually relax. But that's why we like age beef, to make it more tender, just to give it a little bit more time for those connections to be broken down, and for it to taste better. Sorry, Dr. Obscuri, making a weird face.
You're making me hungry, if anything.
Okay, good.
All right, I'll go ahead and close this poll for you in 5, 4, 3, 2, 1.
Right. Fantastic. So yeah, those multipolar neurons are going to be the most common type of neuron. So if we look through all of the different types, we have these unipolar neurons. So we have just one process coming off this cell body here, and then it's going to split into two. We're going to call both of these axons. I don't know why. I didn't make this stuff up. Someone else did it. So both of these are going to technically be axons. This is going to be the cell type that we're going to find in that ganglia that Dr. Frank showed us earlier, that dorsal root ganglia. So these are going to be really associated with sensory. Bipolar neurons have one process that's an axon and one process that's a dendrite. These are really very specialized. We're going to find them in places like the retina, and then in the olfactory epithelium. These are definitely the least common type of neuron. And then multipolar, again, really our most common type of neuron. We're going to have this cell body with multiple dendrites and all of these little dendrite branches, and then one single axon that's going to come and branch here. So a lot of these in the central nervous system, these are going to be both sensory and motor information. So information is going to come in here through these dendrites and then travel down this axon to be communicated. If this was a motor neuron, you might have a skeletal muscle fiber at the end of this. It can also be neuron to neuron communication as well. Great. All right, so moving on to the cardiovascular system. What layer of the heart here is labeled with an asterisk?
All right, we will close this poll in 5, 4, 3, 2, 1.
Finally got you on this one. So let's go through the different layers of the heart, okay? So here in this middle one, this big thick layer, that's the myocardium. That's the part that's going to have a lot of cardiac myocytes or cardiac muscle in it. The endocardium is the inner layer. So I guess let me do some orientation. So this is inside the heart. So we're looking at this directly pushed out. So this would be the ventricle. This is the outside of the heart. So the endocardium is the innermost layer. It's going to look really similar to the inner lining of blood vessels. We've got a simple squamous epithelium here, an endothelium, okay? And then we have epicardium, which is here on the outside. That's that outer layer of the heart. If we add on the pericardium, okay, this layer can end up with a couple different names, which no one really enjoys, but here we are, right? So this epicardium can also be called the visceral layer of the serous pericardium. Okay, so the pericardium is a connective tissue layer, right? That's going to be surrounding the heart. We're going to have a few different layers of that. So we have the fibrous pericardium, and then the parietal layer of serous pericardium, and then we have this pericardial cavity that's going to be full of this serous fluid, so that when the heart expands and contracts, these two layers, this epicardium or the visceral layer of the serous pericardium and the parietal layer of the serous pericardium, really slide very nicely across each other, so that with each expansion and contraction of the heart, we get this really nice movement and no friction here in between. So the epicardium can be considered part of the pericardium. Yes. Yeah. Loving making things confusing. All right. Nice. So again, here we've got this middle layer. That's our myocardium, really thin endocardium on the inside, and then epicardium, connective tissue on the outside. All right, so what is the most superficial layer of the epidermis?
Alrighty, we will close this poll in 5, 4, 3, 2, 1.
It's great. So here we're looking at the layers of the epidermis, right? If we zoom in a little bit more, we can see all of these layers. The stratum corneum is the most superficial layer, so that's the outer layer that's facing the air, right? These are keratinized cells, so this is a keratinized stratified squamous epithelium, right? Underneath that is going to be the stratum lucidum in thick skin mostly. Then we've got this stratum granulosum that has those keratin granules in it, and then stratum spinosum, and then stratum basale underneath it. So remember, the stratum basale is where we're going to have that cell proliferation, and the cells are going to migrate upward and eventually fall off or desquamate that stratum corneum. All right, what type of epithelium lines the trachea?
And this poll will close in 5, 4, 3, 2, 1.
Okay, fantastic. So this is a pseudostratified columnar epithelium with cilia, right? So we can see this pseudostratified columnar epithelia here, beautiful little cilia here on the top. We've got some goblet cells there going to make some mucus. If we were down in the bronchi, like lower bronchi, bronchioles, then we would have a simple columnar epithelium with cilia. So the type of epithelium here in the respiratory tract will change as we get into smaller portions there. But here in the trachea, we've got a pseudostratified columnar epithelium with cilia, also called a respiratory epithelium. All right. Where else would you find respiratory epithelium?
Right. And we'll close this one in 5, 4, 3, 2, 1.
Like my distractors weren't all that distracting. So here, right from the trachea, we're going to get down to these main bronchi and segmental bronchi. As we get down into the bronchioles, that's where we're going to start seeing this change, this shortening of the epithelium, and movement from a respiratory epithelium into a bronchiolar epithelium, which is more of a columnar epithelium. And then down in those terminal and respiratory bronchioles, we're going to start seeing some alveoli coming off, and that is going to be a simple squamous epithelium. All right. Identify this layer of the gut tube indicated by the asterisk. I'll give you a hint. This is the lumen.
All right, and close this one in 5, 4, 3, 2, 1.
Ooh, I gotcha. Stumped you. It's good to know what you should review, right? So starting here at the lumen, right? So we're going to go to the more superficial layers to deep layers. So this first layer is going to be called the mucosa. The mucosa is going to have multiple different layers in it, depending on what type of organ that we're looking at. So here in this particular mucosa, we've got a simple columnar epithelium. We've got some lamina propria underneath, which is made up of loose connective tissue. We've got a muscularis mucosa, which is made up of smooth muscle. So this is all mucosa. And then deep to that is submucosa. So that goes about here. That's a dense irregular connective tissue. And then we have muscularis, which is this big outer layer. So we're gonna have an inner circular layer and then an outer longitudinal layer. That's all made up of smooth muscle. And then here on the outside, we have a serosa. I only know that because I know that this is from the small intestine, right? The portion of the small intestine that is taken from is going to have a serosa. So understanding this layer of the gut tube, really just layers of an organ is something that, again, my medical students struggle with. So keep working, keep reviewing the content, and you'll definitely get it down. All right. What is the function of the myometrium of the uterus?
And you guys are doing really good with this one, so close it a little bit earlier. We'll close it in 5, 4, 3, 2, 1.
Awesome. Fantastic. So one thing that hopefully helped you a little bit was myocardium earlier. That myo means muscle, so that prefix, right? So that myocardium had cardiac muscle in it, right? So this myometrium of the uterus is going to be the layer that has smooth muscle in it. And that's going to be the one that contracts during childbirth. So the uterus has 3 layers. So myometrium is this middle muscular layer. We also have the endometrium. The endometrium is going to be the part of the uterus that's facing the lumen. And this is where we're going to see implantation and development of the placenta and embryo. And then the outside of the uterus, this layer is the perimetrium, and that's going to help reduce friction. The uterus is going to change in size just a little bit, just through regular cycling, just due to the hormones. So estrogen will build it up a little bit, and then you'll have sloughing off of that inner lining, that endometrium. It's going to shrink down a little bit more, but Obviously, the biggest that the uterus is going to get typically under normal circumstances would be if there is a developing fetus in there. And again, that muscular layer of the myometrium, these smooth muscle cells are in all different directions. So it's really going to help that uterus contract down all the way to push that baby out. All right, and that is all I have. I did take some of these review slides from the tutoring session, so feel free to go back and review those in certain sections that maybe you didn't do as well and as you wanted to. All right.
Thank you so much, Dr. Mapes. That was awesome. And again, wish that you had taught me histology the first time around, and I think I would have done a lot better that first time. All right, so it's my really great honor to introduce our last presenter. That's Dr. Natalie Yoshioka. So she is also a faculty member at the Pacific Northwest University of Health Sciences in Yakima, Washington, with Dr. Mapes. So not only are they both presenting here today, but they're also colleagues. Also, it's been such a great honor to know Dr. Yoshioka as we went to grad school together at Penn State, along with Dr. Haggerty. And so not only is Dr. Yoshioka a triple threat in that she knows anatomy, histology, and embryology, but she's also a phenomenal educator, a researcher, and a medical illustrator. So she's really just has done such a great job in the scientific community, and I'm just so grateful that she's taken the time to come and teach you guys some embryology tonight. So without further ado, Dr. Yoshioka, if you want to present your slides.
Awesome. Thank you so much, Mary. Okay, let me share my screen. Can everyone see this okay? Okay.
I see your
Can
right
you
now,
see the poll?
right now I see your, oh, there we go. Now I see your slides. I can't see
Okay.
any polls.
Okay, amazing. Can you see the chat if I share my screen like this? I just want to make sure I don't miss questions.
No, I'm just seeing your presentation. I think you're good.
Okay, amazing. So hi everyone, I'm Dr. Yoshioka. I'm an assistant professor of anatomy here at PNWU, and today we'll be reviewing some embryology concepts. So the objectives that I'm basing these questions off of, they can be found at the Anato-Bee website. And so they kind of go in order. And oh, sorry, the chat. So for the first section, we'll just go over some general terminology, and I have the corresponding learning objective listed for you here. A young female patient reports that she missed a period, as her last known menstrual period was 8 weeks ago. She explains that her periods typically last 5 days. A home pregnancy test taken yesterday indicates that she is currently pregnant. If the test results are confirmed by the physician, what is the gestational age of the embryo?
Right, this poll will close in 5, 4, 3, 2, 1.
Okay, so the correct answer is B, 8 weeks. And so the embryonic or fetal age, that begins when the sperm meets the egg. But it's really difficult to know exactly when that happens. So a better measure for fertilization is to use gestational age, because most individuals will know when their last known menstrual period was. So gestational age is calculated from that first day of the last known menstrual period, because at about after 2 weeks after that last known menstrual period, ovulation will occur, and that is when the individual is most fertile. And so a follow-up question for this could be, what is the embryonic or fetal age of the embryo? And so the answer would be 6 weeks, right? So for gestational age, you're adding 2 weeks to the embryonic or fetal age of the embryo. Okay. So then second question, which of the following results from meiosis 1 of spermatogenesis?
Okay, we will close this poll in 5, 4, 3, 2, 1.
Okay, so this one is kind of tricky. So recall that when we're looking at our precursors, the spermatogonium and the oogonium, those are going to be dividing by mitosis. So we're not changing any chromosome number at this point. So they divide and they form primary spermatocytes, or in the case of the formation of a mature egg, the oogonia are going to undergo mitosis and form primary oocytes. When the primary spermatocyte or the primary oocyte undergoes meiosis 1, that's when they form secondary spermatocytes or a secondary oocyte. And so here, the genetic material is separated. So here, the homologous chromosomes, so the pair from both the mom and the dad, are going to line up on the metaphase plate and separate. So now we've separated our diploid cell and we've formed 2 haploid cells. Or in the case of oogenesis, we've formed a secondary oocyte and a polar body. And then after that, the sister chromatids will separate to generate the more mature form of the sperm, the spermatid, or the more mature form of the egg, the mature ovum, right? So as we move from a primary to a secondary spermatocyte, that's when meiosis 1 occurs. And to move from a secondary spermatocyte to a spermatid, that's when meiosis 2 occurs. And if we look at the contrasting events during oogenesis, moving from a primary oocyte to a secondary oocyte is by way of the process of meiosis 1. To move from a secondary oocyte to a mature ovum, that process occurs by meiosis 2. So kind of tricky, because you would think spermatogonium to primary spermatocyte would be the result of meiosis 1, but no, it's just mitosis. Okay. So next question. Following fertilization, the zygote is blank and has blank number of total chromosomes. Oh, to answer the question, spermatid to sperm cell is just by a process of maturation. So it's just changes in the morphology of the sperm cell. So that happens after meiosis 2. Good question.
All right, and you guys are quick with this question, so we will close it in 5, 4, 3, 2, 1.
Perfect. Okay. So yeah, exactly. So here when the mature egg meets a mature sperm, now we have two pairs of homologous chromosomes in a single zygote. So this would be called a diploid cell or a diploid zygote with 46 chromosomes. So 23 homologous pairs. Excellent. Okay. Next question. The first primary germ cell layer to be differentiated during early gastrulation is the...
All right, this poll will close in 5, 4, 3, 2, 1.
Okay, awesome. Yes, the answer is endoderm. So during week 2, we have a bilaminar disc composed of an epiblast and the hypoblast from the inner cell mass. And then the epiblast forms this indentation called the primitive streak that's going to allow cells to migrate from the epiblast and displace the hypoblast. When that happens, that forms the definitive endoderm. Then that process happens again to form the definitive mesoderm, and then the layer left over is the ectoderm. So endoderm forms first, then ectoderm and mesoderm. And then the ectoderm or the notochord will form in the center of the embryo. That's the central organizer, which will then induce the formation of the neural tube. Excellent. Okay, so next question. Which of the following is formed from the endoderm?
Okay, this poll will close in 5, 4, 3, 2, 1.
Dude, you guys are killing it. I'm like very impressed because I know some of our med students would not be able to get this question either. Liver, yeah, amazing. So during the 4th week of embryonic development, now we're moving from a trilaminar disc to a 4-dimensional structure by the process of lateral and craniocaudal folding. So the ectoderm will form the outer surface or the epithelium, as well as the neural tube. Then the mesoderm will form paraxial, intermediate, and lateral plate mesoderm. The paraxial mesoderm will form the somites, the intermediate mesoderm will form the urogenital structures, and the lateral plate mesoderm splits into a splanchnic layer covering the internal organs, as well as a parietal layer that is lining the internal body cavities. As well, the splanchnic layer also forms limb bones and the limb bud. So we can eliminate that answer. And then as this ventral folding process occurs, it's going to pinch off the endoderm internally to form an internal tube. So sometimes this is called a tube within a tube. This endodermally lined structure is going to form the gut tube, which runs from the mouth to the anus. So that eventually is going to form the esophagus, the stomach, small intestine, large intestine, whatnot. And then the accessory organs of digestion are going to bud off from the gastrointestinal tract. So that will form the liver, gallbladder, and pancreas. So liver is the correct answer. The placenta is formed from the outer cell mass, which differentiates into the cytotrophoblast and the syncytiotrophoblast. So it's not that. The amniotic cavity will form from the epiblast, and then the serous membranes covering organs, that's formed by the splanchnic layer of lateral plate mesoderm. Great work. Okay, next question. Anencephaly, a defect in which a baby is born without a brain, results from
This poll will close in 5, 4, 3, 2, 1.
Okay. So the ectoderm is going to form the neural tube, and you can see that the neural tube has been formed completely here. But what is happening is the neural tube will invaginate internally to form a groove that then closes off. And so that will form this tubular structure that runs dorsally. So here in this sagittal section, you can see the neural tube that's extending from the caudal region to the cranial region. Caudally, it's going to form the spinal cord. Cranially, it's going to form the brain. So if you have a failure of the posterior neuroport close, that will result in spina bifida. If you have a failure of the anterior neuroport close, that results in anencephaly, which is a baby born without a brain. During this process of neural tube closure, you can see that these cells that are on the superiormost portion of the neural crest or the neural groove, these are neural crest cells. And when this tube fuses, and it kind of fuses like a two-way zipper, these neural crest cells will lose their adhesions to the neural tube, and then they can migrate to other parts of the body. Okay, does that one make sense? Okay. All right, next question. Improper neural crest cell migration may result in defects of which of the following?
Okay, and this poll will close in 5, 4, 3, 2, 1.
Okay, so we're kind of split here. If I move to the next slide. So we're kind of split between aorticopulmonary septum and the spinal cord. So the spinal cord will be formed by the neural tube, and the neural crest cells are the cells that emerge after the neural tube fully fuses. So brain and spinal cord neural crest cells aren't involved with anymore. So the neural crest cells are migratory and they will go to so many places. The mnemonic for these neural crest cells and what they form is motel casa. So they form melanocytes, odontoblasts, tracheal cartilage, enterochromaffin cells, laryngeal cartilage, craniofacial structures, arachnoid and pia mater, Schwann cells, spiral septum, and all ganglia. So for example, dorsal root ganglia. So you can see that they're involved in the formation of many structures. I'd like to highlight craniofacial and heart structures because there are a couple of defects in which neural crest cell migration is affected, and you'll see characteristic signs or symptoms will present as craniofacial defects in concordance with heart defects. So the aorticopulmonary septum is the septum between the pulmonary trunk and the aorta, and it spirals because that allows for the spiral orientation of those two great vessels. And that's why it's called the spiral septum. And the neural crest cells are important for creating this septum. The thyroid gland is going to emerge as a structure from a pharyngeal pouch. So it's not this guy. Limb musculature is derived from the splanchnic or the paraxial mesoderm, the spinal cord by the neural tube, and then the kidneys intermediate mesoderm. So aorticopulmonary septum, just in general, if you see craniofacial defects with heart defects, you should be thinking neural crest cells. Okay.
Just so it's on the recording, can you just go back and go
Yeah.
back to that slide? Let's just rewind and talk about thyroid gland versus parathyroid gland versus thymus gland. Let's just clarify that.
Oh, sure. Yeah, the thyroid gland is going to be involved. It's going to be located anterior to the trachea. It kind of looks like a butterfly, and it's going to be involved in secreting hormones that regulate metabolism as well as blood calcium levels. Parathyroid glands are also involved in.
Watch carefully.
Sorry? Oh,
I think
parathyroid
you're okay.
glands are posterior to the thyroid gland. There are 4 of them, and those also secrete hormones that regulate blood calcium levels. The thymus is a structure that matures T cells, so it's involved in the immune system, and it's located anterior to the heart, mainly during the earlier stages of life. Does that help? Was that a question in the chat?
No, but I think you had said thyroid gland was associated with pharyngeal apparatus, and it's not really, right? We have to have the development of the.
Oh, I'm sorry. Yes.
Yeah, we
Yeah.
just want that to
My
be
apologies.
really
Yes.
clarified for everyone.
Yeah. Yeah, my apologies. The thyroid gland is associated with tongue development, not pharyngeal pouches, to clarify for the recording. Thank you for that, Dr. Peterson. Okay. So next question, which of the following are formed from the somites?
All right, and we will close this poll in 4, 3, 2, 1.
Okay, so we're a little split here, but that's okay. So the somites develop from paraxial mesoderm, and the paraxial mesoderm can be divided or differentiates into the dermatome, myotome, and sclerotome. So these root words or prefixes, dermatome, myotome, sclerotome, will help you to determine or remember what they do. So the dermatome, you can see it's just deep to the ectoderm, which forms the outer epithelium. So that forms the dermis and subcutaneous tissue. The epidermis, again, that is derived from ectoderm. The myotome will form the musculature of the body. So that forms musculature of the trunk as well as limb musculature. Then the sclerotome, you can see that they're located more medially and they're going to surround the neural tube. So this forms the bones of the vertebral column and ribs. So the bones of the limbs, arms and legs, that's going to be formed from the splanchnic layer of lateral plate mesoderm. So bones of the limbs we can eliminate. Smooth muscle will be formed mainly from splanchnic layer of lateral plate mesoderm, and then the epidermis will be formed by the ectoderm. Okay. Okay, next question. What structures are formed by the primary heart field?
Okay, and final answers in. Close it in 5, 4, 3, 2, 1.
Okay, so this one is kind of tricky. The primary heart field develops the initial endocardial tubes, or the initial heart tube, and I like to think of it as the inflow part of the heart tube. So the singular heart tube has blood coming in through this inflow part and blood coming out through this outflow part. The inflow part, you can see that with the development of the secondary heart field or the lengthening of this heart tube, this inflow part swings posteriorly to form mainly the right atrium and left ventricle, right and left atria, and then left ventricle. This outflow part will form the right ventricle and then the outflow tract, which will form the pulmonary trunk and the aorta. So left ventricle is the answer. The atrioventricular valves are formed by endocardial cushions. Pulmonary veins will grow into the heart. Interatrial septum will develop after the looping of this heart tube. And then the right ventricle is formed by the secondary heart field. Yes. Okay, what is the distinction between primary and secondary heart fields? So the primary heart field will develop first. The secondary heart field lengthens the heart tube at this outflow part. Okay. Is there another question? I saw a hand. Yes. Yes.
I did have a question.
Yeah.
I didn't know if I was supposed to wait or not.
Oh, yeah, no worries.
For that question, that particular question, I
Mhm.
thought it was the interlateral septal, because I was thinking of maybe like septal disorders, like, and congenital heart diseases when infants have atrial septal defects or ventricular. That's kind of what I was thinking of, and so.
Yeah, so that's going to develop after the primary and secondary heart fields will create these looping events. So you can sort of think about primary and secondary heart fields as forming the chambers of the heart, and then septation events as separating the chambers of the hearts, with the interatrial septum, interventricular septum, and then atrioventricular valves. Does that make sense?
Mm-hmm.
Okay.
Yes, ma'am. Because at first I was like, man, I should know this.
Yeah.
Art. And I was like, wait, that's kind of hopeless, but thank you. Yes, ma'am.
Okay, perfect. Good question. Great question. Okay. So we're gonna skip this question since it's another cardiovascular system question and it's a little tricky. We'll move on to, so the couple of the rest of these are respiratory system, gastrointestinal system. Let's just do the most difficult content because you guys are familiar with gastrointestinal embryology. So let's go to urinary and reproductive system since we're just running short on time. Renal agenesis results from defects in which of the following structures? Renal agenesis means failure of a kidney to form.
All right, try to get your final answers in. This poll will close in 5, 4, 3, 2, 1.
Okay, awesome. So the definitive kidney is going to be formed by the metanephros. So the pronephros and the mesonephros, those are the initial stages of kidney development, and those will degrade. Only the metanephros will persist. So the metanephros will emerge as a bud that contacts the metanephric blastema and induces the formation of the kidney parenchyma. Perfect. Okay. We'll skip this question. All right. The Wolffian duct develops blank while the Mullerian duct develops blank. So we'll do this question and then one more question.
All right, you guys are doing a good job with this one. We'll go ahead and close it in 5, 4, 3, 2, 1.
Okay, so those of you that put A, you're on the right track because Wolffian duct does develop male structures. Mullerian duct does develop female structures. However, the testes and the ovaries are the gonads, whereas the ducts are supporting structures. So the Wolffian duct will develop into the epididymis, ductus deferens, seminal vesicle whatnot. The Mullerian duct will develop into the uterine tubes, uterus, and vagina. The rectum and urinary bladder develop from the cloaca, which is the end of the gut tube, and then the uterus and ductus deferens, if we swap these two answers, then that would be correct. But again, Wolffian duct develops into male structures. Mullerian duct develops into female structures. The AKAs for these, Wolffian duct is the mesonephric duct, and the Mullerian duct is the paramesonephric duct. Okay, so then last question, Treacher-Collins syndrome impacts development of the first pharyngeal arch. What structures do you expect to be affected?
Alrighty, and please get your final answers into the final question of the night. Close it in 5, 4, 3, 2, 1.
Nice. Okay, so if we take a cross section through this proximal most part, the pharyngeal arches or pouches, arches, and clefts, they're going to develop structures. I like to think of it as the lower half of the face and neck. So the clefts are formed from ectoderm, the arches are formed from mesoderm and neural crest cells, and the pouches are formed from endoderm. And so the arches are the real meat of the pharyngeal arches. Those form mainly musculoskeletal structures of the face. Pouches will form mainly glands like tonsil, thymus, parathyroid glands, cells of the thyroid gland, and then only the first cleft will persist as the external ear. There are 5 in total, numbered 1, 2, 3, 4, and then 5 regresses and 6 remains. So the first pharyngeal arch will develop into the mandible and muscles of mastication, as well as upper jaw structures, etc. Awesome. So for the sake of time, we'll end there. Any questions? Okay, awesome.
Thank you so much again, Dr. Yoshioka, for sharing your, again, expertise and again, just some really great questions there to help them apply that knowledge. So really appreciate that. And really awesome job for the students tonight. I am just wowed at how well you guys did. I could tell that there's a level up even from last year that we saw, sort of in both in participation and in just the way that you guys have just engaged the material. So awesome job, everybody. I did see one question in there. Dr. Peterson, did you want to take that one on?
Just very quickly, everybody, we wanted it on the recording. The question was really about, well, what happens after we take the virtual exam? How will we know how we did? Will we be able to immediately or at some point be able to see what the correct answers versus our wrong answers were? And the answer to that is actually no. What you will get from us, because this is going to be a virtual competition that you're submitting your answers through a link to a Google Forms exam document, your answers will be submitted and graded automatically. And then Drs. Haggerty, Mapes, Pescura, and I will send out in individual emails to each of you just what your score was and whether or not that score allows you to qualify for an invitation to the regional competition. And there will be a follow-up email if that is the case for you. We will not show you the exam because some of those questions each year get recycled or modified for the following year of competition. So we do want you to know how you did. We're just not going to show you any of the exam questions, not the correct answer or the incorrect answer. So I just wanted to make sure everybody understood that. Anything to add, Dr. Proskura?
The only thing I was going to add is just another question that we always get, and that we also won't be telling you what your ranking is amongst the other participants. We know that this is a competition, but we want the focus for you to be more about what your performance was, and so we won't share that information either.
A follow-up question just very quickly, and then Sammy will get to you. So how many people advanced from the local level to the regional level this year? And I said something at the end of our last tutoring session. Let me just give you an update on the numbers. We now have over 560 high school students across the U.S. registered to take this virtual competition exam. Now, some things may come up and maybe not everyone will, in fact, but we're hoping that you all do, will go ahead and sit for the exam. Approximately 100 of you will, in fact, qualify to move on to the regional level of competition. So we like those odds a lot. I hope you like those odds a lot. All right, Sammy, what is your question?
So that was actually a part of it. I was gonna say, did it kind of grow some with the participants in the competition? And the answer is yes to that. Oh my goodness. Okay. Okay. I'll hit the books. But okay. All right. 500. Wow. Also, this was really fun. And the questions, as soon as you got them right, you got the adrenaline rush. I loved it. So that was really fun. But I will start studying now again. Because wow. Okay. But thank you. Yeah.
Perfect. Thank you, Sammy. And I see a question about location for the regional competition determined yet. We do know, and we will publicize the information to you guys shortly once we get just a couple more final details ironed out, and we'll post it on the website and send you all an email as well. I also see, since the test is virtual, is there a certain time we have to take it at, or is it any time throughout the day? So that one is, we will be having it at an exact time during the day, depending on when your time zone is. So again, it will be, if you're starting in Pacific time, it will be at 9 a.m., Mountain Time 10 a.m., Central Time 11, and Eastern Time noon. And so you will have to attend during that session according to the local chapter coordinator that you're paired with. So as of right now, again, if you are getting the emails from the Anatomy about this tutoring session, then you definitely know that we have your registration. If you're unsure, feel free to either re-register or send us an email and confirm that you've registered. But as of right now, all you need to do is keep studying and wait for that email to come in by this Friday, and they'll provide you with the rest of the information that you need. And then the final question here is, I see, is the people moving on to the next level per each local competition or just nationally top 100 scores? Trying to understand how.
I think they're trying to ask, are there a certain number of allocated seats in each region for those that are moving on? And I'll let you go ahead and answer, and then I'll throw in my 2 cents worth.
Okay, so what we had done previously was we did have some from each local chapter from last year. We did that is how we did that. So as far as I know, then we are going to continue to do at least one from each local chapter, and then we would be looking at the numbers of participants in each local chapter to determine how many from each local chapter would then be accepted. All right. Well, that's all that we have. If you guys have any other questions or if anything's unclear, again, feel free to reach out to us by email. Feel free to reach out to your local coordinators if you have already been in contact with them. And again, best of luck to everybody. Thank you again to our co-hosts for the night, Doctors Frank, Mapes, and Yoshioka, again, for all of your help in writing some really awesome questions for them to review. And thank you, participants, for doing such a great job of just participating and engaging this entire tutoring session season, and then especially again tonight. So we've really enjoyed all of it, and it's been so great to see you guys grow in this knowledge and show just a really heavy interest and passion for it. So that's all I have, and I hope you guys have a wonderful night, and we will see you at the competition in less than 2 weeks.
Thank you. Bye.
All right. Thanks, everyone. Bye-bye.